After hearing about the different aspects on the consumer part on our first session, on this second session, we would like to move on into the production side and to consider what are the available tools we have in the toolbox in order to allow and generate this healthy, nutritious food we were just describing. And so it's a great honor to have here as our keynote speaker today with us, Professor Kevin Folta. He's from the Horticultural Science Department, University of Florida. He was for the last 6 years the head of the department. We'll hear today about solutions of world food security, availability, but not used. Thank you, Kevin, please.
Okay, thank you. That's great. Now, I'm going to do something a little different. Let me do this first. Okay, there I am, right? I don't have my glasses on, so it's really hard to see. Oh, here it is. Okay, so the first thing you'll notice is that the title doesn't match the one in your program. And the reason for this is simple. I wanted to do, I wanted to get feedback and listen to everybody and learn about what we might be interested in doing before tailoring the final talk for this particular event. And so I did some changes and some remodeling because I would like to make sure we're all on the same page. You see on the program, GMOs. What does this really mean? And what does it mean in the broader context of the way we change plants genetically? So I'm going to use a very crude survey tool. We're going to use applause as a survey tool. But rather than do applause, because sometimes somebody on one side will be very, like Miss America or the Queen, another person will be way too enthusiastic and it skews the data. So what I'd like to try to do is when you agree, and I go like this, I want you to clap one time. Okay, so let's try it. Ready? Okay, the largest per capita population of scientists and engineers are in Israel, and that's the best we can do. Okay, let's try it again. Ready? Perfect. Okay, so we'll use this as a tool as we go through the talk today and try to understand, because what I'd like to illustrate for you is how much misunderstanding there is about this terminology. Okay, and hopefully get us all on the same page about what plant genetic improvement really is. So my research, I'm a researcher. I work in strawberry flavor and the genomics of strawberry flavor. My terminal degree is in molecular biology, but we study also how light works to change the content of food and the quality of food and the flavor of fruits and vegetables. But I won't talk about that today. Instead, we'll talk about genetic improvement. And I come from Florida, which has a lot of similarities with Israel with respect to our horizon of different fruit crops and vegetable crops. We're a horticultural state. We don't grow a lot of corn and soy and big agronomic crops. But we grow a lot of specialty crops, and my department breeds a lot of specialty crops. So we're very interested in genetic improvement. How do we make better fruits and vegetables that are more resistant to disease, perhaps have better nutrition? A whole series of different things that we breed for, disease resistance. So why do we do it? Why do we bother to improve crops? Whether it's in Florida, Israel, or across the developing world, anywhere. And really it's because we need to help farmers. We need to help them manage plant diseases, fuel costs, labor, less expense. We need to minimize our environmental impact. We need crops that can grow with fewer resources, especially water and fertilizer, with sensitivity to the environment. We can help consumers in the industrialized world. We heard already today about the wrong food choices. And how do we help people make better food choices because the quality of the fruits and vegetables are better, they taste better, or maybe last longer after harvest. This way we can ensure that those calories are actually consumed. And expanding food options, particularly for those in need in the developing world. How do we ensure that they have food that can be fortifying to their deficiencies and help that malnourishment problem that David talked about earlier today? So to do that, I'll share with you 3 different stories that are very important in ways in which crop genetic improvement can affect 3 very important situations. This boy is 12 years old. He's in Yemen, and he's receiving on his tongue drops of beta-carotene, so the orange pigment in carrots. And it's suspended in oil, and he receives several drops a few times a year, if he's lucky, to be able to save his vision and also save his health, save his life. Vitamin A deficiency is a problem worldwide. So if we have crops that can be more fortified in beta-carotene, the precursor to vitamin A, we can help ensure food security and health security throughout the world. This is one good example of how crop genetic improvement can play into that, and Dr. Lowe will talk about that later today. This is the citrus disease that's ravaging Florida. And I noticed beautiful trees here in Israel. This is what ours looked like about 10 years ago. Now, most of our citrus industry is infected, and most of it is dying. New innovations in disease resistance, whether through traditional breeding or other modern techniques, are desperately needed. I had the opportunity to visit Uganda last year and really learned a lot about the traditional food choices. And the Mutation banana is a centerpiece of every meal. Yet this type of food is threatened by disease and also doesn't have enough vitamins to sustain the visual health. Again, vitamin A deficiency is rampant in the region. So these are 3 stories we'll come back to at the end. But what are ways that crop genetic improvement can help us attain those goals? And what I would like to do now is walk you through all the different ways in which we change crops in order to be more suitable to solve these kinds of problems. Okay, so these are the problems that we'll... So we know that plant genetic improvement can have lots of different roles, both increasing yields, allowing plants to grow in different conditions, safer products, all kinds of aspects about plant genetic improvement have made crops more suitable for human consumption. But it wasn't always that way. I should say before that, there's a lot of pushback against crop genetic improvement. People say, well, I don't want you to play with genetics. I want it natural. I just want it the way that nature left it, the way that people found it originally. The problem is that most of our crops come from places that aren't currently where we grow them, right? So if you look at where the major crops are grown, you find that quite a bit does come from the Fertile Crescent, from this area of the world, but quite a bit comes from somewhere else. Most of our fruits and vegetables are coming from South America, a little bit from North America, but oranges and citrus from Southeast Asia, apple from Kazakhstan. So there's many different crops that have been taken from their original environments, extracted from their original soils and their original ecosystems that are now been moved to these artificial environments we call farms. So right away we see that what we grow on a farm is not natural. It's not a natural thing to farm. We're taking plants from where they belong and moving them to where we find them most useful. We also have changed the genetics of plants substantially based upon our needs. And here you can see the precursors to corn, banana, or tomato, and their modern varieties. And humans did this. This wasn't natural. This was done because humans did selection. They picked the best one of each generation and advanced those seeds to the future. We've only really been able to accelerate this process of selection in the last 50 years. Before that, it was a very slow process, and still is for the most part. So, how do we change plant genetics? And that's the major theme of the presentation today. How do we do it? And with that, we can ask some questions about what is the best way to do it, what ways make the most sense for our given application. Plant sex. Plants cross with each other. They sometimes self-pollinate depending upon the type of plant. But it really is a question of moving pollen from one part of the plant to another. And plants do this naturally, certainly by pollinators like moths and bees, but also by wind and other processes of physical transfer from one place to another. Some plants self-fertilize even before the flower opens, right? And there's a lot of this that's natural, but humans can intervene in this process. Traditional plant breeding. Traditional plant breeding is really a process where humans change the architecture of the flower or physically move pollen from one place to another. And the idea is mixing genetics in ways that we can find favorable outcomes. So how many people think this is natural? Ready? Okay, so a few people think this is natural. Okay, you'll get it. How many people think that this would be considered GMO? Okay, okay. How many people feel that this is genetic modification? So you can already see the problem we have here just with our terminology and vernacular. How can we all be on the same page about food choices for food security when we don't even have a common vocabulary? I mean, you really see how this is just plant sex, moving pollen from one place to another. And what's really interesting is that we have no idea what genes we're affecting. We're affecting 40,000 or more genes in ways we don't understand. It does happen in nature. We don't know anything about the risks or the impacts. We have no idea. Fine, we eat it, no big deal. Nobody wants to ban it. Maybe it'll take 50 years to achieve a new variety. We also do this thing called inbreeding, where we can cross a plant with itself over successive generations to make a plant that's very genetically uniform. And what's really neat is when you take an inbred plant that's been bred against itself for many generations, and you mix two of them together, sometimes you get a really favorable outcome. So you take two plants that you breed against themselves for years and years and years, and then you mix them, then you finally cross them together and get a very strong outcome. And you can see the effect of this over on the right. These are yields of corn in the US that until 1920 they were relatively flat, but then when we started to make these hybrids, you saw this take off in terms of its productivity per acre. So how many people think this process is natural? Okay, so crossing a plant with itself over and over again and then taking those plants and mixing them together is considered natural by some people. Okay, no problem. Is that considered a GMO? And really the answer is no, that's not considered a GMO. You can sell it in an organic market, nobody cares. But we have no idea what genes we're changing. We're shifting 40,000 or more. What's really interesting, a paper came out several years ago that said 5,000 genes are different between this one and this one. 5,000 genes different. We don't know what most of those genes do. Do they encode toxins? I don't know. No big deal. Nobody tests it. Nobody cares. We can make wide crosses to try to bring genetics together that normally wouldn't come together. Strawberry is a great example. The one grandparent from strawberry comes from Chile. The other one comes from North America. And they got together in a French botanical garden in the 1700s. So here this was this mixture of genetics that happened because a spy brought them from Chile and colonists brought them from North America. They got together in a French botanical garden, and someone realized this made a nice product. Really cool story. But is that natural? It is. I don't think that. I mean, it took a spy and a ship to bring it back, and this is human facilitated. But lots of genes affected. Nobody cares. It's no big deal. We have, no one ever assesses the risks. It's not a problem. We eat it, and it's wonderful, and I love strawberries. The big problem with plant breeding is that we have this problem called linkage drag. Like say you have one plant that has a gene that makes it resistant to disease, and another plant that has outstanding characteristics, but it's very prone to disease. If you cross the two together, you get a plant that's really not, maybe not good for anything. That maybe it gets disease, maybe not. But so what plant breeders have to do is what's called backcrossing, which means you go against one parent while still keeping the trait. So let me just illustrate this by talking about chestnuts. The American chestnut was destroyed back in the late 1800s by a fungus. Destroyed a huge range of trees. And people have tried to bring it back by crossing it against the Chinese chestnut, which is immune to that fungus. The problem is shown here, that if you cross the American and the Chinese chestnut, you get something that's 50% American, 50% Chinese. So then you cross that one against the American chestnut, you look for the disease resistance, so you bring in the Chinese chestnut resistance gene. But now cross that back against the American one, you get something that's 3/4 American chestnut, 1/4 Chinese chestnut. Then you do this again and again and again. You can see how long this process would take to be able to restore mostly American chestnut genetics, but keep that resistance gene. This might take hundreds of years. They're working on it. But you can see nobody really cares. We're mixing lots of genes. That's fine. We can do neat stuff in the laboratory by making crosses that used to be thought to be impossible, because we can make crosses where the embryo is not terribly viable, but then be able to rescue that embryo by putting it in the right kind of media and having that plant grow. And it's able to do this. We're able to make some very, very wide crosses between plants that normally could never cross together. We're able to combine genetics that normally wouldn't happen. And of course, again, I could say, is this natural? No. I mean, we're in a laboratory with forceps pulling apart seeds. It's not very natural. But we don't know. We never test it. Nobody really cares. You can grow it, have it in organic cult production. Nobody wants to ban it. No big deal. We don't know how many genes are affected. More than 40,000. Polyploids. This is really fun. So all of us probably have in each cell one set of chromosomes from mom, one set of chromosomes from dad. Okay, we're diploid. We have one set from mother, one set from the father. And most plants are this way as well. But plants do something funny. Sometimes they have pollen or an ovule that has too many chromosomes. And you get this phenomenon called polyploids. And this occurs naturally. But it also occurs when we want it to occur. We can use different chemicals to induce polyploidy, to induce what we call non-reduced gametes to be able to get tetraploid plants. And the reason that we like this is because tetraploids tend to be bigger. And this illustration on the bottom shows the diploid branch versus a polyploid branch. It makes bigger leaves, usually bigger flowers, and bigger fruits. Sometimes those fruits are sterile. So they don't make viable seeds. At least I should say that. So watermelon's a great example. That it's typically a triploid. It has 3 sets of chromosomes. It has, you look at wheat, has 6 complete sets of chromosomes. So our normal bread wheats are very complex. Here we're transferring an entire chromosome, whole set of chromosomes. We're mixing whole sets of chromosomes. It does occur in nature. We don't know about adverse effects. We don't test it. We have no idea, but we're mixing large sets of chromosomes. So is this a GMO? No, it's not. It's not considered GMO. But is this genetically modified? You can see why the language is a problem. I neglected to mention earlier that in traditional breeding, just by mixing pollen with ovules, there have been cases of adverse side effects seen. We've been able to breed celery and potatoes and a few other crops which actually have accumulated large levels of toxins. So this idea of what's natural sometimes can be very dangerous. Plants do make nasty compounds to protect themselves. And sometimes we can breed those all into one line. And I neglected to mention that, but it's a very important point. So mutation breeding, this is really cool. You can take seeds or flowers or other plant parts and use radiation or chemicals to damage DNA. And once in a while, you make a good trait. How cool is that? And you can make high lycopene in different citrus fruits. A lot of disease resistance traits are developed in grains using mutation breeding. You would be surprised at how many plants that you consume every day, plant products, come from mutation breeding and damaging DNA with chemicals or radiation. So this one, we have no idea how many genes are affected. It could be tons of them. We could have many, many damages per gene. We just don't know. Does it happen in nature? Yes, when mutation happens. We don't know about health effects. We don't test it. Nobody really cares. You can grow it in your organic production in the States. You don't have to label it. No big deal. How many people think this is natural? Not really, right? Is this a GMO? Wait for it. All right, so it is something that people debate, right? To me, this is the most profoundly random human-induced genetic change. a total mixing of the genetic deck in ways that we have no idea what's happening. All we know is that we get a trait that we like. We do other things by combining the nuclei of two cells from two different species. You can put these together in one nucleus. That happens in citrus. They're able to do that, and it makes very nice trees, some of which are resistant to disease, which is really great. But when we talk about this one, transgenics, and this is when we start to use what's called genetic engineering. This is where you can use bacteria to transfer a single gene that we understand into a new genetic background. And this is what's really interesting about this, is that this is what we normally do think of as GMO, or genetically engineered. A couple of good examples of some traits that have been engineered are resistance to insects, so that we can cut pesticide use, increase of shelf life by having plants that may be products that don't brown or stay good looking longer. Also be able to engineer, say, that vitamin A pathway, that beta carotene pathway, so that it would accumulate the orange color. That's also been achieved by traditional breeding, and Dr. Lowe will talk about that later today. But this kind of, the big difference between this kind of technique and others is that you really affect a few genes that you really understand. They test for collateral effects. We haven't seen adverse health effects, some environmental risks, but they're not necessarily specific to genetically engineered plants. They're tested for health risks, tested for environmental impacts. You can't grow those in organic production. People want to label it, but you can go very fast. So what's really interesting about this is that you can see when we know precisely what we're doing, people get nervous. When we have no idea what we're doing, people don't care. And to a scientist, this is really curious. But some crops have been genetically engineered. I kind of want to scoot ahead to the punchline here. We can do this other kinds of genetic engineering where we use and you'll see this term sometimes, instead of transgenic, you'll see cisgenic, which means the gene comes from itself. So putting an apple gene into an apple. And this was done using apple scab, or to solve the problem of apple scab, whereas it took traditional breeders over 50 years to do this by breeding. They were able to do it in 5 years by taking the gene from the wild apple and moving it into the cultivated apple, just the one gene. And again, they know exactly what gene is affected. And basically it's an apple gene in apple, just that you moved it by the laboratory rather than moving it through plant sex. And again, this is one that people feel should be labeled. It's not acceptable for organic cultivation. They are tested rigorously. The new technology is called gene editing. And the new technology will be used to make designer changes in specific genes. So whereas the other one, you're transferring an entire gene and all of the hardware that goes with that genetic change, in this case, you're able to make a discrete edit. So whereas transgenic means you're going to the library and putting a new book on the shelf, gene editing is going into the library and erasing a letter from a book. And we can think of a lot of good examples of books that if you just changed a few letters, you changed the entire meaning of the book. Okay, so there's it's a really important distinction to make. This technology is thought to be able to revolutionize medicine and agriculture. This one, we know exactly what gene we're affecting. We know that it doesn't occur, at least in plants or animals in nature. We know that you're able to very easily assess collateral effects, and that's been demonstrated very well in the last few years. However, we have no idea how this will be regulated. That's still up for debate. There are many people who say it must be labeled, it must be banned, it must not ever be used. We don't know what it's going to do. You hear this all the time. Other people say this is the secret to rapid crop genetic improvement that will be huge for agriculture and tremendous for medicine. So somewhere in between. But the important part here too is that we're having this debate about is this safe or is this something that needs to be regulated when we know exactly what gene we're affecting. We know what that gene does, yet people are a little bit nervous about this. However, if we put a plant in a radiation source and we pound it with radiation, we damage genes in no way we have any idea what they're doing, that's okay. This table is a pretty famous table now. I made this back in 2012, but it's been downloaded about 1 million times. It didn't have gene editing on it, but you can see that this is a comparison of all these ideas and techniques. That traditional breeding, you're moving a lot of genes. You don't know necessarily what you're doing in terms of what genes you're moving. You just know you get something that you like out of the other side. You can patent this, no problem, and obtain IP protection. But it takes a long time. Traditional plant breeding, even for something like tomatoes, something that's annual, may take 5, 10, 20 years to get all the genes in the right place. Hybrids, same idea. Polyploids, we can double chromosomes, and nobody gets too upset about it. You can damage DNA with mutation breeding. You have no idea what you're affecting. All you know is that you get better quality traits. And nobody gets too worried about it. No one knows if there's any adverse effects. The surprising part to me as a scientist is that when we incorporate the precision of being able to very carefully move a single gene or affect a single gene, now we start to see public controversy. You start to see that when we know exactly what we're moving and we're testing it, and we assess what the gene does and assess if the product is safe, that these are the technologies that people are most nervous about. And these are the ones that people would like to ban, or these are the ones that people feel shouldn't be moving forward. So the big question is I really want you to think about that. As folks, and I wanted to do the test in the beginning about who did science, like biology, and then who did social science, and I really would like to know that in a way. But wherever your background is, and however you view genetics as playing a role in global food security, I really want you to think about the inherent contradictions and the intellectual inconsistency of our current approach to regulation and our current approach into how we genetically improve plants. It's really important that if we're going to deliver the best solutions and the most precise solutions, most rapidly to the people who need it, that they should be allowed to have access to these innovations. That the problems of the young man in Yemen, the citrus trees in Florida, or the bananas in Uganda, all of them would benefit from the most rapid technologies being able to deliver the most precise solutions. Yet they're extremely slow to get to the market and the people who need them. So just to kind of wrap it up, humans have always participated in plant genetic improvement. We know that traditional breeding is extremely valuable. Even though we're not sure what we're doing genetically, we just know we like what comes out the other side. Other methods have been used that are extremely random and do catastrophic genetic change. Yet there's no regulation, no interest in regulation, and no fear around those products. Modern plant genetic improvement, we target specific genes of known function. They're tested. We know what the genes do. We can test to see if they're doing what they're supposed to do. Yet these are part of public concern. And I just want you to think about that. It's a really important take-home message as we move very quickly into the future. And as we start to have genetically improved products for people and global food security. So if you have any other questions or you'd like any other conversation around this, please reach out. And I do a podcast every week. And if you'd like to know more about these particular topics, I really do urge you to listen. We have 140 very interesting episodes. Thank you very much for listening.
Thank you, Kevin. We have one question.
Okay.
Room for one question.
A room for a question. Way in back, yes. So mention the use of microorganisms during the process. Yeah, so microorganisms, so gene transfer is typically facilitated by bacteria that naturally do this as part of their life cycle. Scientists have been able to harness that capacity and use it to specifically introduce gene sequences of interest. So that's done everywhere from transgenic, cisgenic, and even CRISPR-Cas9 gene editing. The gene editing process has, whereas it's different in that old school genetic engineering, transgenic and cisgenic, leave behind some of the hardware of that microbial transfer. The CRISPR-Cas9 system allows very discrete changes to be made, and in some cases not leave anything else behind other than that discrete genetic change. It's the same thing that could happen by mutation breeding or by natural variation, but you can make the decision as to where that change occurs. So. Good.
Yes,
Can we expand that a little bit and comment about the prevalence of bacterial genes in our genomes in general?
Yeah, so that's a really important point. So we have entire organelles in our cells that originate from bacterial symbionts. So meaning that we have a tremendous number of genes in our own cells, a large number, that are bacterial in origin and very similar to bacteria. And a number of other genes of very similar function to those that are found in bacteria. We all do share some basic metabolic functions, especially, well, I don't want to get into the details, but there are some basic pathways that are shared by many different organisms. So it's not surprising that we would find similar genes in genetics. So.
Thank you very much.
Also.